Evolutionary trees are a common, if not universal, part of all scientific papers involving evolutionary biology or organismal biology in general. What we're going to start off with talking about here is being a reasonable user of evolutionary trees so that we understand the messages that they're trying to convey about evolutionary relationships. One of the original commands given to humankind, if you buy into this kind of thing, was to name the animals. Now, this is a job that's been taken on by natural historians from time immemorial, and certainly from the time of Aristotle and the other classic thinkers. By the end of Aristotle's work, there was this understanding of this ladder-like set of relationships of all living things, with simpler, more elemental substances along the bottom, and then progressively becoming more and more complicated up to the divine on the other end. And this was been kind of Latinized into what's called the scala naturae, or the great chain of being. Now, this kind of ladder-like thinking is common for many beginners when thinking about evolutionary biology, but it doesn't really represent the way that we understand the genealogical relationships of living things that we try to do with more modern methods. In the 1700s, Carl von Linné worked to try to codify the scala naturae through his work in analyzing the parts of living things. The way that he went about his work was to look for characteristics and then to describe the traits of these characteristics of different living things. Linnaeus worked mostly with what we would today consider to be homologous characters. Homologous characters are parts of living things that we think have the same shared developmental evolutionary history. So in this case, where we're looking at the bones of mammalian forelimbs, we know that they are all different shapes or different functions, but they're constructed of the same elements. So for characters that are related to the humerus, the yellow bone, or the radius and ulna, the next two bones that come along, we can compare the shapes, what the traits are of those characters across these groups in order to make interpretations about how they evolved. So for systematic work today, we do our best to find homologous characters so that we can compare them across different groups of living things. Now, what's not a homologous character? There are other ones we call analogs, analogous characters. So there are four kinds of flying animals, at least there have been in Earth history. So of course we have bats, birds, pterosaurs, which are the flying reptiles, not dinosaurs themselves, though they're related to them, and insects. Now, even though they all have wings, those wings are not constructed by homologous parts. So often what we will see is that ecological demands can craft similar whole body shapes like wings, but develop them out of different bony structures or different muscular attachments. The vertebrates are at least similar in that they all have homologous bones, but although the proportions are different in each. The bee, on the other hand, is completely non-homologous because it's not constructed out of bone at all, and it's just a very different way to be able to build up a wing. The overall function is the same, but they're made by analogous parts, not by homologous ones. I know. Everybody knows the Linnaean hierarchy. This part isn't hard to understand or particularly controversial, but the thing to remember, I guess, is that Linnaeus identified a series of nesting categories, a hierarchical system where smaller groups, what are called lower taxa, like species and genera, are enveloped inside higher taxa, larger taxa, like orders or phyla. But one thing that we need to bear in mind when we're doing big kid phylogeny, ones that are kind of shoved in between, we're going to see how those work in a little bit more detail in another video. But for now, understand that it's not that simple 8-level taxonomic hierarchy that we all learned in high school. There are many higher taxa that we use to label up evolutionary trees to relate different characters that have been acquired or different characters that have been derived in species or in populations. The thing that happened after Linnaeus's work with homologous characters, however, is that he really didn't find evidence of these ladder-like relationships among living things, but rather saw this branching hierarchical kind of system. So we started seeing more visual representations of relationships of living things in more of a tree format. One way to look at Haeckel's tree here is you see that groups will branch off of other groups, and they'll tend to be kind of classified along the side by like level of organization. Back in the day, we used to call those grades, like perceived level of body complexity, which is a term we really don't use today for reasons I'll get into later. There is one figure in The Origin of Species, and it's this one. In this figure, Darwin is representing a number of different groups along the bottom with letters. And what he's showing is the earliest level there where you see the letters happening. And then the Roman numerals along the top from 1 to 14, that's showing different time slices. Now, this is kind of an expandable view of relationships of living things. The way that I would propose that we think about this diagram is Darwin's basic thought about this vision of a tree of life, where we can talk about individuals or populations or species that are related by blood. Today we'd think of being related by genes, but it's a similar kind of thinking. There's a long tail to get into this. Today, evolutionary biologists use a system of naming and organizing taxa that we call phylogenetic systematics. It's a method called a cladistic method, where groups that are more similar to one another in particular characteristics are going to cluster together closer on a tree. Now, and this basic method was developed in the 1950s to 1960s, first articulated in 1950s in German, then translated in English in the 1960s. And over the next 20 or so years, biologists basically came on board from this old sort of grade level of thinking of like ecological organization to character-based tree thinking, where we're looking at more quantifiable differences among living things to establish relationships. So just some terminology you need to have. We're comparing different groups. In this case, we're looking at species. These groups are called taxa, just like taxon is the basic of taxonomy. Each of these taxa are grouped together at the end, so we call these the terminal taxa of the tree. Along the way, there are places where these branches will split. They split at places called nodes. Nodes represent hypothetical ancestors of the taxa that are at the terminal taxa that are at the end of the tree. These 4 terminal taxa make a particular kind of group that we call a monophyletic taxon, or we also call it a clade. Monophyletic taxa are all of the descendants of that hypothetical ancestor that I pointed out at that particular node. Right above that, there's another monophyletic group, the one between tuatara and wall lizard, and also the mammals up at the top from human to platypus. Those also form clades. So these are the kinds of relationships that we're looking to understand. Natural groups that are related by genes. Some other terminology. When you have 2 species that are one another's closest relative, we call them sister taxa. The anole lizard and snake are also sister taxa, as is the seagull and kiwi. In addition to those groups that we're more interested in, I guess, there are also other groups for reference put in called outgroups. Those outgroups represent what are the character states of more distantly related living things. However, here on this tree, the thing to understand, at the very root of this tree, basically at the bottom where tuna and shark is, even though we might think of tuna and shark as being more primitive compared to like humans or snakes, all of the terminal taxa are equally related to that most recent common ancestor. In addition to common names that we would give for different types of groups, we can also add on higher taxa onto our tree. For example, Humans and opossums both give live birth. We lump them together into the infraclass called Theria within mammals. And so, but if you include the platypus too, all of those are mammals together. The class Aves, it represents the living and fossil birds. The birds and all of those other things that we might think of as reptiles, we group together as diapsids. One last thing, as I've noted at the bottom here, all these trees are generated using an R script that I found online that was very helpful, and you are free to please do search for that if you're looking for a good work through for starting to learn to use evolutionary trees. A little bit about the difference in the appearances of these evolutionary trees. The one on the left is what we call a cladogram. The branching pattern is the same as the phylogram on the right, but the phylogram, the branch lengths indicate degree of evolutionary difference, whether this is put in terms of nucleotides in a DNA sequence or in terms of whole body morphological characteristics. Another way to be able to represent these taxa is through a chronogram. So the x-axis now is millions of years, and you notice that some of those groups, Brontosaurus, Velociraptor, Archaeopteryx, Confuciusornis, don't make it all the way to the end with the living taxa. That's because these are extinct ones. So it allows us to hypothesize when particular groups branched off from one another in terms of geologic time now. When we read an evolutionary tree, we'll notice a few different kinds of groups that mean different things. The kind of group that we're always looking to try to recover are groups that we call clades. Again, monophyletic groups that are joined by a shared derived trait that we also call synapomorphy. So all of the species that are boxed off here within Aves, they all share one single common ancestor at the node with Archaeopteryx, and that includes all of the descendants of it, both the extinct ones as well as the living species. And again, what always defines clades are inherited, shared, derived traits. Shared primitive traits aren't helpful for being able to recover these. So for example, here, a classic group that we always talk about with like air quotes today is a group that we call reptiles. So the reptiles that I've boxed off on this tree includes living reptiles as well as some extinct Mesozoic dinosaurs. The problem is that this doesn't include all of the descendants of that hypothetical ancestor at the bottom of the reptile node. So we call this a paraphyletic group, particularly because it doesn't include all descendants, and because what they share are ancestral traits. If we wanted to make these more natural biological groups to turn them into clades, we would have to include those extinct Mesozoic dinosaurs in with their descendants, the birds. So oftentimes when people talk about dinosaurs, they're talking about the big Hollywood dinosaurs from the Mesozoic. But when systematists are talking about dinosaurs, that includes those big extinct ones as well as living birds. So we always say that birds are living dinosaurs, but we see birds kind of nested within diapsids here. So the larger group isn't reptilia. The larger group is diapsida, which has to do with cranial architecture that's going to unite all of the terminal taxa in this boxed off area. Another kind of group, I just kind of made this group up because I couldn't come up with a better one looking at this tree. If I wanted to lump together the kiwi and the human because they walk on two legs and they can't fly, and I want to call this group bipedalia, that's weird. That's not like a natural group. They're not really connected by genes. The poly part meaning that it's coming from different ancestors. So in this case, we see bipedality as a recent independently evolved trait or a trait reversal rather than a shared derived characteristic. So to summarize this, if we are looking for natural groups that we call clades, monophyletic groups are what we're trying to aim for, where we have taxa that are joined by an inherited synapomorphy. The other types of groups, polyphyletic and paraphyletic groups, are not natural groups, so systematists tend to avoid using them whenever possible. Certainly in writing, although informally we might talk about reptiles, but that's the same kind of thinking that goes into like that grade level of thinking of like ecological similarity as opposed to genealogical similarity. So what we're really going for here is we are trying to make a family tree, not like a pedigree, but a family tree that shows relationships based on genes. We're not always able to fully resolve a tree, however. I boxed off the clade at the bottom to say that that clade is fully resolved, meaning that each pairwise set of relationships has been sorted out. On the other hand, above it, the unresolved part, where it's kind of a comb, what this means is those 4 kinds of monkeys make a clade, but we don't exactly know which ones are sister taxa to any of the others. This will happen oftentimes if you're working with lots of characters when you don't get like a consensus result on what the appearance of the traits are for those taxa. It's not the end of the world, but it's just something to know that it does happen. A few things that are common for beginners to miss when interpreting evolutionary trees. First of all, forget about ladders, okay? There's no higher and lower. All living species are modern ones, and all living species that are shown on any tree are equally related to the most recent common ancestor at the base of the tree. Next, avoid reading the tree tips from left to right, or in this case, bottom to top, and assuming that the ones at the right or the top are more advanced. Just because we're people doesn't mean we're the pinnacle of creation, because that's ladder thinking again. So there's no teleology here, meaning that there's no purpose-oriented evolution when we're looking at natural history. And third, don't interpret clades with only a few nodes or a few members as being more primitive. It's usually more about sampling. So if you look at this, the turtle looks kind of lonesome there all by itself, or the tuna, whereas the clade of dinosaurs that looks actually pretty deep. That doesn't mean that those groups have more advanced characteristics about them. What it means is that it just has to do with, what that means is that those are about the choices that we made when we were sampling to choose the taxa that we wanted to compare in the first place. Another important use of phylogenetic systematics is to be able to see how characters evolve over evolutionary time. Here we're looking at feathered flight. So you can see at the node where Archaeopteryx branches off, we see Archaeopteryx, Confucius ornis, and the seagull as all using feathered flight. Now we know the kiwi is also a bird and also has feathers, but it doesn't fly. So in this case, we see the loss of this character. So we call this a homoplazy, when we have the reversal of a character from an initial state to a more from one state to another one. When we design evolutionary trees, we have to bear in mind what's important about the characters too. In order to know which ones we should pick, there are a lot of questions that go into this. We have to know which ones are stable over evolutionary time. Think of the presence of fur in mammals. Most mammals have fur, but whales don't, humans don't. There are a few others that don't have extensive fur on them. Maybe they'll have a few hairs here and there. But using like a heavy pelt as a characteristic can be a little bit fraught, because it's going to end up causing some interpretations to go a little squirrely on you. If you can, use lots of characters. This can be tricky if you're doing morphological characters. It's easier if you're working with molecular data, particularly if you're figuring every base pair position is its own character, which we'll deal with later. Because you can at least go with the majority. Even if your tree ends up not being fully resolved, you should be able to get at least the general consensus outline of what the relationships is among your taxa. And then last, use effective outgroups for reference to determine the polarity of a character. Characters can have different traits, say either the ancestral or primitive one, or the one that's derived from that ancestral state. By using effective outgroups, we can know which the ancestral state is with a good degree of confidence. That's the main stuff I wanted to get through for using evolutionary trees today. The next step is going to be about putting them together.